Hello everyone, welcome to my channel. From ancient farming practices to cutting-edge biotechnology, agriculture has come a long way, but today, powerful innovation is at the center of global debate. Genetically modified crops. Are they a revolution for food security or a risk to biodiversity? In this video, we will dive deep into the world of genetically modified crops, how BT cotton was created using Bacillus thuringiensis, the objectives and application of genetic modification, the regulatory landscape, its advantages, disadvantages, and why India has not approved any new genetically modified crops since BT cotton. Stay with me till the end as we explore how genetically modified crops can shape the future of agriculture in India and across the globe. What are genetically modified crops? Genetically modified crops are plants whose DNA has been altered using genetic engineering techniques. So we are using recombinant DNA technology, that is also known as genetic engineering technology. The process involves introducing new genes or modifying existing ones to produce desired traits or to produce desired characteristics. So what is the global adoption in use? Genetically modified crops were first introduced in the United States of America in the year 1994 with the flavors fiber tomato, which had been genetically modified to slow tomatoes ripening process, delaying softening and rotting because this genetic modification was already in use to produce insulin, human insulin, that is humulins, different types of vaccines, antibiotics, and drugs on a large scale. So current status about the genetically modified crop is that recent data from the International Service from Acquisition of Biotech application shows that more than 18 million farmers in 29 countries, including India, planted over 190 million hectares of genetically modified crops in the year 2019. So genetically modified crop is a in which we are altering the gene and the new and inserting the new DNA, so we are introducing into the plant cell. So plant genetic modification entails inserting a specific stretch of DNA, a specific piece of DNA into the plant's genome to confer new or different characteristic to give rise to new or different traits. So modification is a technology that involves inserting DNA, a lean piece of DNA or gene of interest into a host organism's genome. The most widely grown genetically modified crops worldwide are cotton, soybean, maize, and canola with herbicide tolerance and resistance. So this is a main objective of creating genetically modified crops are the plants should be herbicide tolerance and insect resistance. Uh, Five top genetically modified growing countries are USA, Brazil, Argentina, India, and Canada. These countries together accounting for approximately 90% of the area of the genetic modified cultivation. Why in the news? The Supreme Court pronounced a split verdict on the validity of the decision of Union government granting approval for environmental release of genetically modified mustard crops in the year 2022. It is about the there is a verdict about the release of genetically modified mustard crops in the year 2022. More on the news, there was a split verdict of two-judge bench. One judge held that approval given by the Genetic Engineering Appraisal Committee in the year 2022 about the genetically modified mustard crop is defective and contrary to the principle of public interest. The judge noted that the Food Safety and Standard of Authority of India has not conducted any study on the impact of genetically modified mustard crop on health, on human health. Second judge held that the approval was not effective. The judge has issued direction for strict monitoring by the union government. Direction passed by the court. The Union of India has to evolve a national policy regarding genetically modified crops. The policy be framed in consultation with all stakeholders, including state farmers, groups, etc. So these are the reason it was in the news in the year 2022 about release of genetically modified mustard crops. Objectives of genetic modified crops reduce dependence of, because it is a genetically modified, so it reduces dependence on pesticide, herbicide as a toxin produced by the genetically modified crops. With the help of, we are producing BT cotton, which is a genetically modified pest resistant plant. It is a cotton variety which produce an insecticide to combat bollworm, a disease which is occur in the cotton. Providing, it provides resistance to certain plant viruses, it provides resistance to tolerance to herbicide, weedicides used to control weeds, it reduces the need to till the soil to control weeds. So there are many objectives of growing genetically modified crops. Now about the, with the help of bt genes, we are making the genetically modified crops. So we have to know about the bacillus thuringiensis. It is a soil bacteria. Soil bacterium Bacillus thuringiensis produces crystal protein called cryproteins that are toxic to the larva of many insects like tobacco, toxic to the larva of many insects like tobacco, budworm, armyworms, beetles, and mosquitoes. The cryprotein exits as an inactive protoxin and gets converted into active toxin when ingested by the insect. So, soil bacterium Bacillus thuringiensis produces crystal protein. The name of the protein is cryprotein, which are that protein are toxic to the larva of many insects like tobacco budworm, armyworms, beetles, and mosquito. The cryprotein exists as inactive protoxin in the and get converted into active toxin when ingested by the insect, as the alkaline pH of their gut solubilizes as the crystal. The activated toxin bind to the surface of epithelial cell of the midgut and create pores in the insect body. This after ingesting it. This causes swelling and lysis of the cell, leading to the death of insect larva. So Bacillus thuringiensis consists of protein, which is name of the protein is cryprotein, which is coded by a gene called cry gene. This gene is isolated from the bacterium and incorporated into several crop plants like cotton, tomato, corn, rice, soybean. With the help of, we are making this BT cotton, BT tomato, BT corn, BT rice, BT soybean, etc. These genetically modified crops having cry or BT toxin genes do not require the protection of insecticide, herbicide, weedicides, as they themselves act as a bioinsecticides. So in this method, we are making the BT cotton and different crops. So Bacillus thuringiensis are soil-tending bacteria that naturally produces a toxin that is fatal to certain insects. Bacillus thuringiensis products several types of toxin. The most well-known toxin is a cryprotein, which is a delta endotoxin. These are crystalline insecticidal proteins which produce during the sporulation period when its spore formation take place. Additionally, it produces another toxic, which is a CYT protein, which are cytolytic and mosquitocidal. Cytolytic means it destroy the cell, and mosquitocidal means it destroy the insect and mosquito. So it is a bacteria, Bacillus thuringiensis. Bacillus thuringiensis are with the help of this, we are making the transgenic crop. We are inserting the cry protein and SIG protein with the help of a direct approach or indirect approach, because it carries a cry protein that are toxic to the insects. So few of the crop we have made BT cotton, BT brinjal, BT corn like this. So when an insect picked on the transgenic plant, the toxic cry protein present in the plant crystallizes the digestive system of insect, eventually leading to its death. However, it has no harmful effects on the human digestive system, even though it has no harmful effect for the plant also. So Bacillus thuringiensis, this cry-BT gene is inserted into crop with the help of any direct approach or indirect approach. Indirect approach, we are using a vector. The name of the bacteria is Agrobacterium tumefaciens. With the help of this, we are introducing into the making the plant transgenic crop, and this crop become a transgenic because it carries a cry protein which is toxic to the plant plant and toxic to the insect when the insect ingests it. So it goes into its gut, mid gut, and create pores. This causes swelling and lysis of the cell, leading to the death of insect larva. So this is the reason, this is the method we are using the transgenic crops with the help of Bacillus thuringiensis bacteria. From the bacteria, we are taking the gene, and that gene is uh, cryproteins. So strain of basis is same like this. It is given here. If the insect swell it or ingest it recognize, it's just bind to the specific site of the gut of the insect, and then the caterpillar stop feeding because there is a swelling happen in the gut, and the pores happen, toxin dissolve, and the bacteria within a 1 to 2 days. That insect, not about the bacteria, that insects who ingest it, the caterpillar of that insect dies from the that cry protein, and as a spores and gut bacteria proliferates in its bud. So this is the reason cry protein is very toxic to the insect, because it create pores in the mid gut of the insect epithelial cell of the mid gut, and this causes swelling and lysis of the cell, leading to the death of the insect larva because of the cry protein, which carries cry gene. So this crygene we are inserting into the any crop and making it transgenic. So with the help of that, we are using two methods. One is the direct approach, other one is an indirect approach. Bacillus thuringiensis is about the Bacillus thuringiensis. Bt gene is transferred Bacillus into the corn, and this insect, if it eats, it will die. So in this way, we are making the transgenic crop, because the Bacillus thuringiensis cannot harm itself. If the plant cannot, Bacillus thuringiensis, that bacteria cannot harm itself from the toxin, because it present in the form of inactive form in its body. But when it insert into the when we are making the plant as a transgenic plants, it is inactive, even though it does not harm to the human also. Whenever the insect ingest it enter inside the after the entering the alkaline pH of the gut, it coordinates with the alkalinity of the, because the gut is the alkaline. So alkaline pH of the gut, this active toxin inactive toxin become active and causes swelling and lysis of epithelial cell, and eventually leads to the death of the insects. For this, we are indirect approach. We are using Agrobacterium tumefaciens as a vector. Agrobacterium tumefaciens is a natural tool for genetic engineer for plants. It is primarily used for making in biotechnology for transferring genes into plant cell. This bacterium naturally carrying a tumor inducing plasmids containing a tDNA region. So this bacteria consists of this bacteria is called Agrobacterium tumefaciens, which is a plant pathogenic bacteria. It infects broad leaves crops including tomatoes, soybeans, sunflower, and cotton. It causes tumor because it has tumor inducing plasmid, or it causes tumor name is a Crohn galls in plants. Tumor formation is induced by, because its plasmid's name is a TI plasmid. It is a Agrobacterium tumefaciens. It is its chromosome and extra chromosome, which is known as a tumor-inducing plasmids, which causes tumor of Crohn in plants. The bacterium transfer, this bacteria is having TI plasmid, has a capability to transfer a piece of its DNA known as a tDNA. Its DNA name is a tDNA. tDNA. It has the capacity to transform the normal plant cell into the tumor cell. It make the plant cell normal, healthy plant cell into tumor cell. So this bacterium is known as a natural genetic engineer. So how we can using it? We can just taking the TI plasmids and from the Agrobacterium tumefaciens bacteria, and then afterward, what we are doing, we use this plasmid from which tumor producing gene we have it has been eliminated. The gene which has been producing tumor in the plant, this gene has been eliminated. and then afterward the transformed bacteria do not cause disease to any crop. Then this ti plasmids we are entering in ti plasmids we are entering the DNA containing gene for desired traits. So this DNA we are taking from the Bacillus thuringiensis, which has cry protein cry gene, which is toxic to the plant. So we are inserted into the ti plasmids, and it's become a recombinant plasmid. So insertion of gene into the tumor-inducing plasmids, but the tumor-inducing gene we have been eliminated previously. So it does not cause any disease. And in that place, we are inserting the DNA-containing gene. That gene is a cry gene from Bacillus thuringiensis, which is toxic to the insects. An insertion of into plasmid using the restriction, cutting with the restriction enzyme and joining with the DNA ligase, it forms recombinant tumor-inducing plasmids. Then we are tDNA carrying new gene with the plant chromosome. So tDNA, the bacterium carries a Ti plasmids which contain tDNA region. This tDNA can be modified to carry specific genes that scientists wants to introduce into the plant, because tDNA has the ability to transform the norm to carry the piece of gene into the host cell. So in this way, Agrobacterium tumefaciens infects a plant cell. It can transfer the tDNA into the plant nucleus where it can integrate into plant's genome. So with the help of this, we are carrying the gene of interest gene we have taken from the Bacillus thuringiensis, which carries cry gene. So this like this we after recombinant plasmids in then it's we are doing the culturing of this tDNA carrying new gene within the plant chromosome, and the plant with new trait will be generated, which are having the cry gene. If any insect will eat it or ingest it, it will die. So in this way we are resulting the plant we are altering the plant's DNA of plant's genome, and the process is known as a transformation, and the resulting plants are called transgenic plants. So it is a image of Agrobacterium tumefaciens. Is bacteria chromosome and extra chromosome in tumor-inducing plasmids. So tumor-inducing plasmid can be utilized to make recombinant DNA after removing the gene that causes gall disease or Crohn gall disease. So we are removing this, and in that place we are inserting the gene from Bacillus thuringiensis cry gene. So the recombinant tDNA is then transferred to the plant using Agrobacterium tumefaciens, which is used as a plant vector. This is the image of a Crohn gall disease, which is caused by the Agrobacterium tumefaciens in many crops like tomato, soybean, sunflower, and cotton. Saying the process is given here, the Agrobacterium tumefaciens, we have taken the Ti plasmids and it carries a tDNA, and then afterward we are tumor producing gene has been eliminated. It does not cause any disease now. Then insertion of DNA containing gene for desired trait. So we are taking that gene is the cry gene from the Bacillus thuringiensis. Then insertion of cutting with the help of restriction enzyme, then inserting into the Ti plasmids, and it become recombinant Ti plasmids, and with the help of ligase we are ligating it, joining in, then introduction into plant cell in culture, and then after the culture, the tDNA carrying new gene within the plant cell chromosome, regeneration of plant, a plant with new trait will generate. In this plant is having the transgenic act as a biopesticide. If any insect will ingest it, the insect will die, because it will go in this plant body, the crygene is inactive, but when it insect will eat it, it will die, because of it become active because of the alkaline nature of the pH of the gut, and insect will die because of the it creates problem in the digestive tract. So Agrobacterium tumefaciens, the same image is given. The plant cell are grown in culture. A plant is generated from a cell clone. All of its cells carry the foreign gene and make space. It as a new plant. The plant become a transgenic plant. So the bacterium is used to insert the tDNA carrying the foreign gene into the chromosome of a plant cell. This is the reason we are using the Agrobacterium tumefaciens as a vector in this case to make the plant transgenic. Now, the same the image is given here. So this is the method of using genetically modified crop to genetically modify a crop. Same the method is given the TI gene of interest into a vector. Vector we are taken, that is Agrobacterium tumefaciens, then TI plasmids taken up by the bacterium in culture, plant cultured with bacteria, then tumor-inducing plasmids move into the plant cell and gene of interest insert in a plant cell. Gene of interest, there is crygene from the Bacillus thuringiensis. Cell is screened for the inserted transgene with the various screening methods are there. Cell transfer to the plant growth medium. So in this way, there is we are making the genetically modified crops. We are using the vector. The vector name is Agrobacterium tumefaciens. In that, we are removing, before doing all the process, we are removing the tumor producing gene has been eliminated. It does not cause any disease, but it carries only the allele piece of DNA which carries a gene of interest, that is a crygene from Bacillus thuringiensis. Other method is the direct method, gene gun method, gene amplification, gene coated on good gold particles, then it suits and then it's a particle at the plant cell and the gene incorporates directly into the plant DNA. So there are many methods are there, direct approach, and then electroporation method is also there, micro injection method is also there. In electroporation method, we are using the as word signifies, we are using electric pulses, electricity, creating pores in the cell membrane. We are making the pores in the cell membrane, allowing the DNA to enter. It is a versatile method and applicable to various cell types and can be used for both in vitro and in vivo experiments. In this way also we are making the transgenic crops. The next method is a microinjection method. In this we are using the fine glass micropaper to directly induce genetic material like DNA into cell cytoplasm or nucleus. So in this way, we are making the transgenic. The most important thing, we are in this method, we are making transgenic animals, which can be used for cell biology research, cloning, and infertility treatments. So advantages of Agrobacterium tumefaciens, which is act as a vector for making the Bacillus BT crop, BT cotton like that plants. So this plant, because it efficiently transfer DNA, it is highly efficient method for introducing genes into plants. Agrobacterium tumefaciens is highly efficient method for introducing genes into plants. It can transfer large piece of DNA, including genes of interest and regulatory sequence. The tDNA, the TA plasmids carries which DNA? The name of the DNA is tDNA. tDNA can integrate into the plant genome, leading to stable inheritance of new traits. The plant is having the new traits. So Agrobacterium tumefaciens is an important tool in plant biotechnology due to its natural ability to transfer DNA into the genome of host plant. So this is the reason we are using Agrobacterium tumefaciens as a genetic tool in genetic engineering. Genetic manipulation of Agrobacterium tumefaciens have yielded considerable advances in increasing transformational efficiency in a number of plant species and cultivators. In this plant is given about the Agrobacterium tumefaciens. In this image, it causes a crown gall disease in many crops, which crops including tomato, soybean, sunflower, and cotton. So this before using this, we are removing this tumor-inducing gene has been eliminated before using it. So application of modified crops, there are many application of genetically modified crops that are being used today. Biofortification. Biofortification is a process. It is a very important process of enhancing macronutrient content. We are increasing the macronutrient content in the food. How we are increasing? The most important example is the golden rice. Golden rice, which is a beta carotene enriched, which is a precursor of vitamin A. Vitamin A. It was the first application of genetically modified biofortification in the year 2000. It also prevent many diseases such as cancer, diabetes, and cardiovascular diseases. So it is the most important application of genetically modified crop, which is a biofortification, because it may prevent many diseases such as cancer, diabetes, and cardiovascular diseases. It is used as an edible vaccine. They offer many benefits than the traditional one due to lower manufacturing cost. The cost is low in manufacturing and much fewer than side effects is also very less. It also uses a biofuels, fourth generation biofuels, which is a renewable source of energy because the feedstock material can be replenished easily. So it used as a biofuels, which is obtained from genetically modified algae and cyanobacteria, blue-green algae, have gained considerable attention. It also used as a phytoremediation. Genetic modification can be utilized to clean up soil and water pollutants. So it is a very important application that it is used to clean up soil and water pollutants by expressing the particular genes that express enzymes dealing with these pollutants. These are the few genetically engineering appraisal committees about. It is statutory committees, and it is a genetically engineered organism or cell in the year 1989 frame under Environment Protection Act 1986. Ministries and composition chairman, co-chairman, representative of Department of Biotechnology, responsibility appraisal of proposals relating to release of genetically engineered organism and product into the environment, including experimental field trials. Other responsibility of genetically engineered appraisal committee is to appraisal of activities involving large scale of hazardous microorganisms and recombinants in research and industrial production from the environmental angle. So genetically about the genetically modified crops in India, the approved crop is only BT cotton. BT cotton is the only genetically modified crop approved for cultivation in India. It can grow on approximately 11 million hectares. Other crops such as mustard, chicken pea, pigeon pea, sugarcane are in various stages of research still now, field trials and deliberation. So there are various regulatory framework in India which is governed by the rules for manufacture, use, import, export, and storage of hazardous microorganism, genetically engineered organism or cells. So these are the advisory, approval, and monitoring committee which are taking care of the genetically modified crops in India. The committee are RDAC, IBSC, RCGM, GESC, SBCC, DLC. India's in genetically modified crops. There is an array of genetically modified crops, that is the brinjal, tomato, meat, chicken pea, which are in various stages of trial in India. Till now, it's going on. There is one of the leading producer of genetically modified crops in the world. India is one of the leading producer of genetically modified crops in the world. There are many regulatory frameworks. There are stringent law in the place to address threats to animal health, human safety, and biodiversity in general during the development, cultivation, and transboundary movement of genetically modified crops. Some statutory bodies on genetically modified crop regulation are RDAC, Recombinant DNA Advisory Committee. Second one is the GEAC, Genetically Engineering Appraisal Committee, and then State Biotechnology Coordination Committee. So RDAC reviews ongoing project involving high risk and controlled field experiments. It also approves the application for generating research information on genetically modified plants. Genetically Engineering Appraisal Committee is responsible for overall evaluation of proposal related to the release of genetically modified organism and as well as the product into the environment. It works under the Minister of Environment. Now, State Biotechnology Coordination Committee, it reviews safety measure in various department. What are the benefits of genetically modified crops? There are many benefits. First one is the battling pests and diseases. Genetically modified crops can be engineered to resist pests and diseases, reducing the need for chemical pesticide, herbicide, weedicide. It is the important things. It is used as a bioengineered product to resist pests and diseases. For example, BT cotton. BT cotton produces its own insecticide. The insecticide, we are inserting the cry gene, cry protein, which carries the cry genes, effectively controlling bollworm infestation. This not only increase yield, but also decrease the environmental impact of farming because we are reducing the use of insecticide, pesticide. In addition, the adoption of BT cotton has led to significant increase in cotton production, making the country a leading global producer. It is also the big advantage. This pest resistance can be particularly crucial in region where crop losses due to pests are a major concern. Second advantage are Weather-proof farming. Genetically modified crops can be designed to withstand extreme weather condition. It can be able to withstand in extreme weather condition, a critical advantage in the face of climate change. Drought-tolerant maize varieties, for instance, can maintain yield under water stress condition. This resilience can help ensure food security in regions prone to erratic rainfall or prolonged dry spell. In countries like Kenya, drought-tolerant maize has shown promise in improving yield during dry seasons. It is also the most important benefits of weather-proof farming. Third, benefit is nutritional. It acts as a nutritional powerhouse fighting hidden hunger. Biofortification through genetic modification can enhance the nutritional value of crops. And golden rice, which we have learned already, that enriched with the beta carotene, aims to address vitamin A deficiency in developing countries. Other examples include iron-rich rice and zinc-fortified wheat. This nutritionally enhanced crop have the potential to combat malnutrition. It decrease malnutrition and micronutrients deficiency, particularly in regions where diversified diets are challenging to achieve. So it is very important that it increase the nutritional, act as a nutritional powerhouse, fighting hidden hunger. Fourth benefit is the green revolution. Genetically modified crops often boast higher yields and improved resource efficiency. Herbicide-tolerant crops allow for more effective weed control, reducing competition for nutrients and water. Crop modified for enhanced photosynthesis or nitrogen use can produce more with less input. For example, research on C4 rice aim to significantly increase rice yield. So C4 rice project aim to improve photosynthetic efficiency in rice and thus to enhance crop yield. So C4 project rice is also very significantly increase rice yields. These advantages could be crucial in meeting the growing global food demand while minimizing the expansion of agricultural land, thereby protecting natural habitats. So, in turns, it protects the natural habitats because we are minimizing the expansion of agricultural land and increasing the global food demand. Fifth benefit is eco-friendly farming, reducing agriculture's footprint. Genetically modified crops can contribute to more sustainable farming practices. Herbicide-tolerant crops often enable no-till farming, which reduces soil erosion, carbon emission. So it also helps in reduction of soil erosion, carbon emission. Insect-resistant crop, it reduces insecticide use, benefiting non-target organism and improving overall ecosystem health. But if we use less use of insecticide, pesticide, weedicide, herbicides, so it will immediately affect our ecosystem. It will improve the condition of our ecosystem. Sixth benefit is self-life superstar. Genetically modified technology can be used to develop crops with extended shelf-life, significantly reducing post-harvest losses. So it reduces the post-harvest losses. After harvesting the crops, there may be many losses. So it reduces the post-harvest losses. The flavor saver tomato, though no longer in production, was an early example of delayed ripening. Extended shelf life could also reduce the carbon footprint associated with the frequent transportation and refrigeration of perishable item, the food which will be renewed very easily. So there will be the frequent transportation and refrigeration of perishable. This could be particularly impactful in developing countries where lack of refrigeration and poor transportation infrastructure lead to significant food loss. So in this way, we are protecting our food. Seven benefits is crops as medicine factories. Plants can be genetically modified to produce vaccine, antibodies, and other pharmaceutical compounds. With the help of this technology, we are developing different types of vaccines, antibodies, and other pharmaceutical compounds. This approach is known as a biofarming. Could potentially reduce the cost and increase the accessibility of certain medicines. For instance, research is ongoing on producing edible vaccine in crops like bananas and potatoes. While still largely in the research phase, this technology hold promise for whole promise for revolutionizing vaccine and drug production. 8 benefits of phytoremediation champions. Phytoremediation is a very important things which we got from the genetically modified crops. Genetically modified plants are being developed for their ability to absorb and concentrate specific pollutants from the soil, which process is known as phytoremediation. Plants have been modified to absorb heavy metals or break down organic pollutants. So we are modifying the plants to absorb heavy metals or break down organic pollutants which are harmful for our soil, which are harmful for our atmosphere, for our environment. So for example, modified poplar trees have shown enhanced ability to clean up contaminated sites. Now, next question is, why has India not approved commercial cultivation of any genetically modified crop since BT cotton? Why has India not approved commercial cultivation of any genetically modified crop since BT cotton? But it is the reason behind this. First one is there is a regulatory hurdles and policy inconsistency. India's regulatory framework for genetically modified crop has been marked by complexity and frequent changes, creating an uncertain environment for approval. So the Genetic Engineering Appraisal Committee, which is responsible for approving genetically modified crops, has often been caught between scientific recommendation and political process. So there is a dilemma between the scientific recommendation and political process. For instance, in the year 2009, Genetically Engineering Appraisal Committee recommended the commercialization of BT brinjal, but then Environment Minister imposed a moratorium. Moratorium is temporary stopping of an activity, especially by official agreement. Moratorium, impose a moratorium, citing the need for more studies and public consultation. So it is also there is a hindrance in giving the other genetically modified crop. This pattern of scientific bodies giving approval followed by political intervention has created a regulatory deadlock. Second reason is public opposition activist influence. Strong opposition from environmental groups, farmer organization, and some scientists has significantly influenced the genetically modified crop debate in India. This group has raised concern about biosafety, biodiversity loss, and socio-economic impacts on small farmers. This is the reason India has not approved commercial cultivation of any genetically modified crops since BT cotton. The case of genetically modified mustard, which received genetically engineering appraisal committee in the year 2017, but remains unapproved for commercial cultivation due to ongoing legal challenges, exemplifies this influences. Next challenge is the economic and trade consideration. India position on genetically modified crops is also influenced by economic and trade factors. There is a concern that widespread adoption of genetically modified crops could affect India's agriculture export, particularly to genetically modified sensitive market like Europe. Moreover, the experience with BT cotton with increasing yield has raised issues but about seed prices and market concentration. The dominance of multinational companies in the genetically modified seed market has led to concern about seed sovereignty and impact on domestic seed companies. Next challenge, the fourth challenge is the political and federal complexities. It adds another layer of complexity to genetically modified crops approvals. It creates another layer of hindrance to the genetically modified crop approvals. While the central government sets over all policy, agriculture is a state project, allowing state governments to have significant say in agricultural decision. This has led to situation where states have banned genetically modified crop trials even after approvals. For instance, in the year 2018, states including Rajasthan, Madhya Pradesh, Bihar, Delhi, Punjab, West Bengal, and Kerala had started their opposition to genetically modified mustard. What are the major challenges related to genetically modified crops? It is across the world. The first one is the environmental concern, health and safety uncertainty. So major challenges which are related to genetically modified crops are environmental concerns. Genetically modified crops raise significant ecological question. This concern about potential gene flow to wild relatives, which could cause super weeds resistant to herbicides. The impact on non-target organism is another worry. It is impacting on non-target organism is another worry. While BT crops reduce overall pesticide use, which we know very well, they may affect beneficial insects also. There may be chances they affect beneficial insects also. Moreover, there is a debate about whether genetically modified crops contributes to biodiversity loss by promoting monoculture farming. So this is also a debate. Second one is the major challenge related to genetically modified crops across in every states, in every country. Health and safety uncertainty. While numerous studies have found genetically modified foods safe for consumption, concern persists about potential long-term health effects. Critics argue that current safety assessment may not capture subtle or long-term impacts. These are worries about the potential for new allergens or changes in nutritional contents. There's worries about their potential for new allergens which causes allergy or changes in nutritional content. So, for example, the Starlink corn controversy in the year 2000, where a genetically modified corn variety approved only for animal feed, entered the human body supply, highlighted the challenges in preventing cross contamination. Next challenge is about the socioeconomic impacts. The adoption of genetically modified crops can have complex socioeconomic repercussions. While they can increase yields and farmer incomes, as seen as a BT cotton in India, there are concerns about market concentration and farmer dependence on seed companies. The high cost of genetically modified seeds and associated inputs can be prohibiting for small scale farmers, can be giving bad effect for small scale farmers. The global dispute over genetically crop patents exemplified by cases like Monsanto legal battles with farmers highlights issues of intellectual property rights in agriculture. Fourth challenge, the regulatory challenges. It is establishing effective regulatory frameworks for genetically modified crops is complex. Different countries have varying approval processes and labeling requirements, creating trade complication. The EU is stringent regulation contrast with the United States more permissive approach, leading to trade disputes. Developing countries often lack resources for comprehensive biosafety regulation. The challenge of monitoring and enforcing regulation, especially in regions with porous borders, add to the complexity. Fifth challenge is the ethical and cultural consideration. Genetically modified crops raise ethical question about the extent of human intervention in nature. They are showing that there is a more extent of human intervention in nature. They are concerned about they are playing with the God, they are playing God, and more complication of crossing. The issue of genetically modified crops also intersects with broader debates about food sovereignty and the right of communities to determine their own food system. This ethical dimension adds layers of complexity to the scientific and economic considerations surrounding genetically modified crops. Coexistence and contamination issues. The sixth hurdle is the coexistence and contamination issues. Managing the coexistence of genetically modified and non-genetically modified crop present practical challenges. It is creating a practical challenges. The managing the coexistence of genetically modified crop and non-genetically modified crops. Cross-pollination can lead to unintended presence of genetically modified material in non-genetically modified or organic crops. In the year 2013, a origin farmer around unauthorized genetically modified wheat in his field, leading to temporary import bans by some countries. Establishing effective segregation practices through the supply chain is complex and costly. This issue is particularly problematic for organic farmers who risk losing certification if their crops are contaminated. Seventh hurdle is the resistant developments. The evolution of resistance in target pests and weeds poses a significant threat to the long-term efficacy of genetically modified crops. BT cotton, initially high effective against bollworms, has been decreased efficacy in some region due to pest resistance. Similarly, the widespread use of um, glyphosate-resistant crop has led to the emergence of glyphosate-resistant weed in many areas. This is the problem also. It's coming. This created technical trade mills, which farmers become dependent on ever evolving genetically modified technologies to maintain yields. What measure can be adapted to promote the balanced use of genetically modified crops in India? Transparent trial sowing seed of trust, establish an online portal where all trial data and results are published in real time, encouraged independent scientists and stakeholders to observe and verify trials. Other method we have to adopt to promote the balanced use of genetically modified crops in India is to biotech bridge fostering public-private partnership. This can help balance profit motives with public interest, establish clear guidelines for sharing intellectual property and benefits. Third one is to green gene bank preserving agricultural heritage. It allocates funding for collection, documentation, and storage of traditional seeds. This initiative can safeguard biodiversity while allowing to for genetically modified crop development. Fourth one, we have to farmer first policy empowering the grassroots, develop policy that prioritize small and marginal farmers in genetically modified crop adoption, create farmer committees at district level to participate in decision making process, support system for farmer adopting genetically modified technology, insurance scheme to protect farmers against potential failures of genetically modified crops. Swift is to eco-impact assessment, cultivating environmental harmony, establish a network of ecological observation to monitor impacts on local ecosystem, develop protocol for assessing effect on non-target organism and biodiversity, implement a system of periodic reviews to assess cumulative environmental impacts. Six, we have to do nutritional navigation and targeting hidden hungers, focus genetically modified crop research on addressing a specific nutritional deficiency prevalent in India. So in this way, we have to collaborate with health experts to identify key nutrients needed in different regions in India, develop biofortified crops tailored to local dietary habits and deficiencies, implement pilot programs to assess the effectiveness of this nutritionally enhanced genetically modified crops. So this targeted approach can demonstrate tangible health benefits of genetically modified technology, potentially increasing public acceptance. So seventh one, we have to regulatory reboot, streamlining with science, to create a clear science-based approved process for genetically modified crops, establish an independent biotechnology regulatory authority, implement time-bound decision-making process, develop clear guidance for risk assessment and management. Eighth one, we have to do label logic, empowering consumer choice, easy to understand labeling system for genetically modified products, educates consumer about genetically modified labeling, is establish strict penalties for non-compliance with labeling regulation, respects consumer rights to information and choice. Ninth one, harmonizing international standards, actively participate in international forums. India must participate in international forum to develop harmonized standard for genetically modified crops. India must work towards establishing mutually recognized safety assessment procedure with major trading partners. It contribute to the development of global best practices in genetically modified crop regulation and trades. By taking a leadership role, India can ensure its interests and concern are reflected in international genetically modified crop policies. So, advantages now, what are the advantages of genetically modified crops? It's better than conventional breeding, space resistance, virus resistant, drought resistant plants, herbicide tolerant, sustainable agricultural practices, enhanced nutritional value. So, better than conventional breedings, it enables genetic modification in such a way that may not have been possible through conventional breeding methods, such as the introduction of gene from different organisms. Pest resistant increases yields, save farmers money, and reduces environmental pollution, save our ecosystem. Virus resistant lack natural resistance resistant. This increases the productivity of crops, drought resistant plant. It reduces the use of groundwater, herbicide tolerance, because the weed removal process such as plugging and tilling is not required for this crop. This also reduces soil, water, and air pollution. Sustainable agriculture practices such as no-till farming, which keeps the carbon within the soil rather than in the atmosphere, enhance nutritional value. This can be beneficial to health and increase food security around the world. Disadvantages of genetic, there are few disadvantages of genetically modified crops. This can lead to the horizontal gene transfer of pesticide, herbicide, weedicide, or antibiotic resistance, thereby affecting the metabolism response to the external environment stimulus of the host or some of the concern of this technology. The first one is that impact, the disadvantage is impact on health. It causes ecological imbalances. It may cause ecological imbalances by causing the spread of diseases among both plant as well as in animals. There may be a chance. Impact of herbicide tolerant crops. Herbicide overuse in field may allow weeds to develop resistance to them. For example, like glyphosate has been used as a herbicide in the United States in the year 1974, and its widespread usage has resulted in glyphosate resistant weeds. Its third disadvantage is impact of pest resistance, as Bt toxin produced by the genetically modified crop, which can overcome the efficacy of this crop. Now, next disadvantage is impact on genetic diversity in crop varieties. It reduces the genetic diversity of neighboring crops, close relatives, and weeds. Next disadvantage is the herbicide-resistant weeds. Weed may also cross with the herbicide-tolerant crops, resulting in herbicide-tolerant weeds, which is more worrisome also. Effect on honeybee and other pollinators. There is a worldwide growing concern. There is a crop may endanger, and the honeybee population and other pollinators, different types of pollinators may also be affected. So these are the few advantages of genetically modified crops. So mandate of ministry and department, there are some ministry of department, Ministry of Environment, Forest and Climate Change, Department of Biotechnology, Ministry of Science and Technology, Ministry of Agriculture, Ministry of Health and Family Welfare, Ministry of Commerce Industry, Central Board of Excise and Custom, Department of Revenue, Ministry of Finance. These are the mandate of ministry department and their function. Status of genetically modified crops in India. Bt cotton only approved. It is only approved to genetically modified crop in the year 2022 for commercial cultivation, widespread infestation. It used to protect the plant from the widespread infestation of cotton bollworm. In the year 2018-19, it was 95% of the total cotton planted in India. In the year 2002, commercialized Bt cotton, which resulted in a significant improvement in cotton production, export, and the textile industry. So it was created to combat the widespread infestation of bollworm. Bt brinjal in the 2009, Bt brinjal was cleared by GESC for commercial cultivation, but it was put on a 10-year moratorium by the technical expert committee following public backlash. So moratorium means the temporary stopping of an activity, especially by official agreement. Recently, GASC has allowed field trials of new varieties Zanak and BSS 793 on indigenously developing BT brinjal in 8 states during 2022-23. So the fruit and borer resistant BT brinjal was approved for the commercial cultivation of GESC in the year 2000, but it was put on a 10-year moratorium due to public outrage and recommendation from brinjal growing states. So there is field trials of new variety of indigenously developed BT brinjal in 8 state form. It's still going on from 2020 to 2023. So BT cotton in India is concerned, but it's about that. It research by University of Manchester show that the effects of BT cotton on farmers yield vary across India states, but most states show a decrease in insecticide use since the introduction of BT cotton. So reduction in insecticide use may be beneficial for farmers' health and environmental by reducing pollutants. There is some concern about the BT cotton in India. By the year 2009, the pink bollworm bollworm had started to develop a resistance to the protein, necessitating further study into genetically modified cotton. So Bt cotton has little effect on cotton yield, and although it may lead to an initial reduction in insecticide use, this effect is now diminishing due to insecticide resistance in insect population. Also, Bt cotton is ineffective against many cotton pests such as plant bugs, stink bug, and aphids. The recent note to pink bollworm resistant genetically modified cotton trials. The new variety of CRY2AI gene, which confers pest resistance on the cotton, is also derived from the bacteria Bacillus thuringiensis. It is a new one. The new variety is a CRY2AI gene. CRY2AI gene. The two gene and the proteins they generate do, however, differ from one another. While CRY2AI targets a wider variety of insects, including some Coleoptera, beetles, and Diptera flying species, CRY1AC primarily target Lepidopteran insects, moth, and butterfly. Significance, what are the significance? Pink bollworm is a significant issue for the cotton industry in India, regarded as one of the production of cotton globally. The cotton yield and quality are decreased as a result of pink bollworm larvae feeding on the seeds and fibers found inside cotton balls. So it's decreasing the cotton yield and quality also. Approval for trials from the GESC could be significant step towards combating this destructive pest. Genetically modified mustard in India has not begun yet. The commercial cultivation of highly genetically modified mustard in India has not started as begun yet. The Supreme Court has recently put a temporary hold on the cultivation of genetically modified mustard. A clear and comprehensive policy to regulate genetically modified organism is still waiting for this. This policy should ensure the safe and responsible development and use of genetically modified technology in agriculture, while also addressing potential risks. So about genetically modified mustard crop DMH11. DM is a Dhara mustard hybrid 11, which is a genetically modified hybrid variety of mustard species. Is developed by Center for Genetic Manipulation of Crop Plants at Delhi University. It paves the way for the commercialization of the country's first genetically modified food crop. Genetically modified mustard has not been released for commercial cultivation yet. DMH11 is a result of crossing between two mustard varieties. First one is a Varuna and second one is the East European early Hira 2. This is difficult to do by conventional breeding because mustard is self-pollinating. That is, the pollen from the male part pollinates and fertilizes the female part of the same plant. The cross has been done after introducing the Barnasa and Buster gene from the soil bacterium Bacillus amylocufaciens. So there are some regulatory framework of genetically modified crops in India. So few regulatory frameworks of genetically crops. First one is the Food Safety and Standard Act 2006. It prohibits import and manufacture, use of or sale of genetically modified food without Food Safety and Standard Authority of India approval. Review Committee on Genetic Manipulations. Third one is the State Biotechnology Coordination Committee. Measure various institutions handling genetically modified organism. Fourth one is district level committee or genetic engineering appraisal committee investigates reports about compliance or non-compliance of regulatory guidelines. Then there is a genetically crop approval process, which it is given on a case-to-case basis after thorough scientific evaluation of health and environment safety as per applicable guidelines made under Environmental Protection Act 1986. So these are few questions of the UPSC PYQ previous year question, pre and mains question. Student kindly go through it. These are the benefits of, in conclusion, there are many benefits of genetically modified methods in agriculture: enhanced yield protection, that is resistance to pest, insects, weeds, and diseases. It reduces cost for food, reduce uses environmentally damaging pesticides, enhance nutritional value. The most important example is golden rice. It provides tolerance to droughts, hence reducing the use of groundwater.